my lovely children. Welcome or welcome back to the Under 10 Minutes series. And today's topic of discussion is the structure of a prokaryotic cell. So without any further ado, let's dive right into it. All right. So this is the structure of a prokaryotic cell that you see on your screen. So let's decode the structure. The outermost layer that you see right here, the outermost layer is referred to as the glycocalyx. Now the glycocalyx has got multiple roles. It is the outermost layer. It's protective in function. It helps in preventing loss of water. So it prevents loss of water, referred to as desiccation. Now it helps the bacteria from the attack of various pathogens like various bacteriophages. Not just that, it also protects the bacteria from the host's immunity, right? Now, if the glycocalyx is hard and if it's a hard and rigid structure, it's referred to as the capsule. But on the contrary, if it's a smooth structure, it is referred to as the slime layer. I hope that made sense. Now, this is the glycocalyx. Now, inner to the glycocalyx, we have the cell wall, right? So the outermost is the glycocalyx, inner to which is the cell wall. Now, if you talk about cell wall, the cell wall in bacteria is made up of peptidoglycan or murine. Now this cell wall, which is actually made up of peptidoglycan or murine, is what actually determines the shape of the cell. So it determines the shape of the cell. Not just that, it also provides a strong structural support. Okay, now talking about the third layer, we had the glycocalyx, then we had the cell wall, now we have the cell membrane. So the innermost layer is the cell membrane, or aka the plasma membrane. Now the plasma membrane in bacteria is similar to that of eukaryotes. Okay, it is also made up of phospholipid bilayer, and this plasma membrane is also selectively permeable. So once again, it is a phospholipid bilayer, it is selectively permeable in nature, and the composition of the plasma membrane of the prokaryotic cell is similar to that of the eukaryotic cell. Alright, now that we've discussed the layer, now let's discuss about the cytoplasm. Now this, what you see right here, is the cytoplasm. Now cytoplasm is the main arena of all the metabolic activities. Now the cytoplasm is the main arena of all the metabolic activities. Cyclosis, or referred to as cytoplasmic streaming, which is nothing but basically the cytoplasm is not stationary. It's in a state of constant motion, referred to as Brownian motion. Okay, cytoplasmic streaming, multiple names again. So cyclosis or cytoplasmic streaming is absent in a prokaryotic cell. All right, now talking about the infoldings of the plasma membrane. Now the innermost layer was referred to as the plasma membrane. Now the plasma membrane, infolds or folds inwards and forms these infoldings or invaginations, okay, which has got multiple names. Now the first invagination, this structure that you see right here, let me zoom it for all of you. This structure that you see right here, this invagination of the plasma membrane is referred to as the mesosome. Mesosome is nothing but the invagination of the plasma membrane. What is the function? Mesosomes increase the surface area. What do mesosomes do? They increase the surface area and the enzymatic content. They help in secretion. They also help in respiration. They help in replication of the DNA, and thereby helping in reproduction, and they also help in cell wall formation. I hope that made sense. Now another invagination of the plasma membrane that you see, not in all bacteria but in certain photosynthetic bacteria, is that of chromatophores. Now what is chromatophores? Chromatophores is nothing but like a pigment storing tubular mesosome. So chromatophore is again a pigment containing tubular mesosome. This is also an invagination or infolding of the cell membrane. Chromatophore specifically you will find in bacteria which perform photosynthesis or photosynthetic autotrophs, right? Chromatophores actually store pigments and thereby helping in photosynthesis. I hope that made sense, right? Now moving on to the next one. Okay, now let's discuss the genetic material that you see right here. Now this genetic material is of the prokaryotic cell or a bacteria is referred to as a nucleoid. Nucleoid is the main genetic material which is double-stranded circular DNA. Nucleoid is a double-stranded circular DNA. It has got multiple names. It is known as the genophore, it is known as prochromosome, it is known as incipient nucleus or nucleoid, right? Now apart from the nucleoid, you also see, I'm not very sure if you can categorically see this structure that you see right here. This is the plasmid. This is the plasmid. Now plasmid is nothing but the extra chromosomal DNA, which is also circular double-stranded. I hope that made sense. So plasmid is the extra chromosomal DNA, which is again circular double-stranded. Now it confers, it confers certain special characteristics to the bacteria like antibiotic resistance or fertility genes, all of that. All right. Now apart from that, you also see the only organelles which are present in bacteria is ribosomes, right? So these structures that you see, these dots that you see, this is nothing but the ribosomes, right? Now, and to be more precise with all of you, the ribosomes that are found in a prokaryotic cell is the 70S ribosome. The ribosomes actually help in protein synthesis. Now ribosomes has got two subunits to it. The larger subunit is the 50S and the smaller subunit is referred to as the 30S. Now S here stands for sedimentation coefficient or Swedberg's unit, okay? Now multiple ribosomes, okay, or like a cluster of ribosomes bound to a single strand of mRNA. Multiple ribosomes bound to a single strand of mRNA is referred to as polyribosomes or polysomes. I hope that made sense. Now apart from that, now let's discuss about the various structures like, let's say, flagella. Now this membranous extension that you see right here is flagella. Now flagella, to be precise with all of you, actually helps in motility or locomotion. So flagella is what actually helps in locomotion. So this is the locomotory structure in bacteria. Now a misconception which masses carry is that cilia is not found in kingdom Monera. Cilia is a locomotory structure that you've witnessed for the first time in kingdom Protista. Flagella is the only locomotory structure which you found in kingdom Monera, right? Okay. Now, talking about the structure of a flagella. Now, flagella has got three parts to it. The first part that you see right here, which is embedded in the cell envelope, right, or the cell membranes, is the basal body. Now, the basal body has got 4 rings. It has got 4 rings to it. Starting from the base to the top, we have first the M ring, then the S ring, then the P ring, and then the L ring. Again, you do not have to go into the details, but just remember that this is the basal body, which has got 4 rings associated to it. And then this structure that you see right here, this is the hook. And the last structure that you see, this is actually the filament. Now, filament is actually the distal end. It's towards the distal end, and this is the longest portion of the flagella. I hope that makes sense. Now, talking about other membranous extensions, we have fimbriae and we also have pilus. Now, what is the difference between the two? First, let's discuss fimbriae. Okay, so the ones that I've highlighted in yellow, these are referred to as fimbriae. Now, fimbriae and pili, they do not actually help in locomotion, right? Fimbriae, on the other hand, they actually help in attachment to various surfaces. So fimbriae helps the bacteria in attaching to various surfaces. So in a way, it helps in adhesion. And these fimbriae are, relatively less in number. They're lesser in number. Okay. Now, talking about pili. Now, these extensions that I've highlighted in blue, these are referred to as pili or pilus. Now, again, these are nothing but these are elongated tubular structures which are actually made up of pilin protein. These are elongated tubular structures made up of pilin protein, and pilus, or sex pilus to be more precise, helps in bacterial conjugation, which is a type of sexual reproduction or parasexuality. Now, these extensions that you see right here, these pili or pili, they actually have no role in motility. They actually do not contribute in motility. They have no role in motility. I hope that makes perfect sense. All right, talking about inclusion bodies, right? Talking about the various inclusion bodies that you see. Now, these inclusion bodies that you see right here, these are membraneless. These inclusion bodies are actually membraneless and they lie freely in the cytoplasm. They are nothing but the reserve food materials. Few examples which are categorically mentioned in our textbook NCERT is the inclusion bodies include, these are nothing but phosphate granules or synophysin granules or glycogen granules. Now, apart from that, the one last structure that we are left with is the ones that I've highlighted in pink here. Now, what is this that you see? This is nothing but like a gas vacuole. Again, it's a membraneless vacuole which actually helps in buoyancy. And with that, we've discussed all the parts of a prokaryotic cell, and I hope you found it useful. In case you did, do share it among your friends and stay tuned and follow for more.